Welcome back to our official YouTube channel, SRN VGNR Gout Arts and Science Degree College, Khammam. Myself, Dr. B. Satish Kumar, Assistant Professor in Biochemistry. In today's class, we are going to learn about the translation process. And before that, uh, we need to have the idea about genetic code. Then only we can understand what is the translation process. For the final year students in semester 6, we have molecular biology paper, core paper. In that paper, we already discussed, you have already learned about the replication process, transcription process, and along with that, post-transcriptional modifications. These three processes you have learned so far, and the next topic in molecular biology, that is unit 2. In this unit 2, we are going to see what is genetic code and what is translation. That means what is protein synthesis, and after that the post translational modifications. Okay, so in this video lesson we are going to learn the genetic code. So that is the first topic, subtopic in this unit, that is genetic code. Let us start the class with genetic code. Here the definition of genetic code is, it is the relationship between nucleotide sequence of mRNA and the corresponding amino acid sequence in a polypeptide chain. In other words, it shows which amino acid is coded by which codon in mRNA. You can easily understand what is the genetic code. Genetic code means, in fact, when we talk about the protein synthesis, that is translation means the synthesis of protein. Protein always has always contains a sequence of amino acids. So to synthesize a particular protein, the organism must use those amino acids in a specific sequence. So that information is present at the genetic level. So suppose in the last class we have already discussed about this. Suppose if we say that one gene is there, gene 1, and that gene 1 is responsible for the synthesis of a protein known as protein X. Suppose if you take this example, suppose we have a gene here, a double stranded DNA molecule, and this gene is having nucleotide sequence that contains nitrogenous bases, ATGs. So during the transcription, what happens from the DNA? A RNA is synthesized, and in case of eukaryotes, that RNA undergoes post-transcriptional modifications to form a mature RNA. So here, suppose it is the mRNA, messenger RNA. mRNA is there. mRNA is being synthesized, which is single-stranded RNA molecule. So now it is also having the nucleotide sequence in it, single-stranded mRNA. From the nucleotide sequence of mRNA, now a protein must be synthesized. That protein contains sequence of amino acids. Okay. So now these nucleotide sequence, the nucleotide sequence in the mRNA will specify their corresponding amino acids. Okay. So one unit of nucleotides, a unit of nucleotide nucleotides may specify one amino acid. So we have 4 different bases but 20 different types of amino acids. So how these 4 different bases codes for these 20 different amino acids? So that is the genetic code. So here the genetic code means the relationship between the nucleotide sequence in this mRNA and the amino acid sequence in the protein or polypeptide chain. So what is the relationship between this one and this one is known as genetic code. So in other words, it can also be defined as which type of amino acid is coded by which type of codon in mRNA. Suppose when we look at the genetic code, okay, so for your convenience I will show you the genetic code table right now. So this is the genetic code table. So in this genetic code table you can see here first one is UUU. UUU codon codes for phenylalanine. So that whenever a UUU sequence occurs in an mRNA molecule, that UUU codes for phenylalanine in the corresponding polypeptide chain. So this is what the genetic code. So genetic code is simply this table. So this table is nothing but it explains which codon codes which amino acid. So that is the relationship between the codon and specific codon and its corresponding amino acid. So that is simply the genetic code. So now in translation what happens? The genetic code is nothing but the translation of nucleotide sequence into a amino acid sequence. So that translation will require the genetic code. So that decodes nucleotide sequence into amino acid sequence. So that is what the genetic code. So as we have already seen the genetic code table, in that we took one example, UUU codes for phenylalanine. So from this you can easily find out some of the features of genetic code. So how the genetic code can be prepared, how genetic code can be read, and what are the features of genetic code we will learn in this class. So the features of genetic code, the first feature is triplet codon. So that means how many nucleotides acts as a single unit to code a single amino acid. Whether it is one nucleotide codes for one amino acid, or two nucleotides together codes for one amino acid, or three nucleotides codes for one amino acid. So that is what the coding ratio, that is known as the coding ratio. So the genetic code is a triplet codon. So as we have already seen in genetic code table, that UUU codes for phenylalanine. So that means three nucleotides, that is U and U. So three nucleotides acting as a code word, that is a code word. So that code word contains three nucleotides. That is what the triplet codon. Why that triplet codon is being utilized by the organisms? If you ask this question, so here you can see the possibilities, different possibilities of codons, the number of nucleotides in codon. Suppose if you take singlet codon, so that means one nucleotide codes for one amino acid. So then what happens? We have only 4 nucleotides that codes only 4 amino acids, but in fact we have 20 different amino acids. So the number of codes must be equal to 20 or more than the 20. It is equal to 20 or more than 20. Otherwise all the different amino acid cannot be coded by these codons. So there must be sufficient number of codons to code 20, uh, 20 different amino acids. Okay, so that is why, suppose if you take a class in which 100 students are there, and if we will allot roll numbers for all the 100 students, so how many digits we can allocate for the students, how many digits we can allocate the students for giving roll numbers for them. Suppose if we use only one digit for allotment of roll numbers for the students, what happens? From 0 to 9 we will get only 10 numbers, so 10 students will get roll numbers and remaining 90 students won't get, because we are using only one single digit. Okay, so if we take two digits, so then starting from 0 0, we can give up to 99, so 100 students can easily be given 100 roll numbers using two digits. So this is decimal system. Okay, in the decimal system we have 10 symbols, but in this case only we have four symbols or four letters, ATGC. Okay, so that is why if we use 4, if we use one nucleotide, then we will get only 4 out of, from 4 nucleotides we will get only 4 amino acids, 4 codons that codes for only 4 amino acids. Okay, if we get 2, if we get 2, that is doublet codon, so we will get 4 to the power 2 number of codons, that is 16. 16 is also not sufficient to code for 20 amino acids. Then we go for 3, that is triplet codon, 4 to the power 3, so that means 64, we will get total 64 number of codons, so that is sufficient to code for 20 different amino acids. Okay, so minimum 2, that 2 is not sufficient, 3 is sufficient, of course it is little bit more, but there is no chance in fact. So either you can use if you want to use minimum number of letters in the code word, then it is only the triplet. So if we use 4, it becomes even more complicated. So that is why the organisms always follows triplet codon mechanism, coding ratio, to keep it simple and sufficient to code for all the 20 different amino acids. Okay, so that is what the triplet codon. So 64 possible codons are there to code for 20 different amino acids. Okay, and the second one is code is universal. So this is you can see the genetic code is same for viruses, bacteria, plants and animals. So different kinds of organisms use the same genetic code table, so that means U codes for phenylalanine. So if I say U codes for phenylalanine, you may ask that in which organism U codes for phenylalanine. So then I will say in any organism U codes for only phenylalanine. So there is no difference in genetic code from organism to organism. So from lower level organism to higher level organisms, all the organisms follow the same genetic code, almost same genetic code. So that is why the code is universal. If it is it may be a bacteria, or it may be a virus, it may be a plant, or it may be an animal, but still the genetic code is same for all the organisms. So that is why it is universal. And third one is code is without punctuation. So that means if you take mRNA molecule and you have suppose A U G C C G A C G, so this kind of sequence is there in mRNA. So then what happens, we take each 3 nucleotides as a single codon. So codon 1, codon 2, codon 3, so in the same way. So that here the code is without punctuation. So that means if we take AUG is the first codon, the second codon starts from the just just the next nucleotide. It will not leave any gaps between two codons. Suppose if we take if it read AUG as the first codon and leave that C and start reading from the next CGA, so that means here it is leaving a gap. So this creates a lot of confusion whether all the nucleotides must be read continuously, or if we have to whether we should ignore some nucleotides bases, or if we have to ignore some bases, which bases must be ignored. So this uh, develops very complications, and so that is why the code is without punctuation, so there is no gaps at all in in a reading frame. So that means each and every nucleotide will be used in the codons, so no gaps will be left behind between the two successive codons, so there are no gaps at all. So that is without punctuation. Okay, so fourth one is non overlapping. So the same thing, this fourth thing is also explained in this earlier case. Non overlapping means each nucleotide is read only in one codon. It will not be read in more than one codon. So that means if we read, suppose if we take the AUG, so AUG CCG AAC, so this is the code mRNA molecule. So this is the first nucleotide, sorry, the first codon, and then the second codon starts from here to here. So that is means this codon is separate and this codon is separate. There is no gaps between these two codons, and at the same time there is no overlapping between these two codons. So just completion of the one codon, the next nucleotide will be the first nucleotide in the second codon. Okay, so there is no overlapping. If there is overlapping, so then what happens? AUG is the first codon, and leaving UGC is the second codon, and GCC. So this way, AUG, UGC, this way. So again, it is CCG. So that means UGC and GCC. So this is the next codon. So that means here if we take that G in overlapping reading frame, so this in the first codon the G is the third base, and in the second codon the G is the second base, and in the same G in the third codon it is the first base. So one nucleotide, one base is being read in three different codons. So that is not the case in fact. So there is no overlapping or overlapping during the reading of nucleotide sequence or codons. So each codon, in each codon the nucleotide, the each and every nucleotide will be used in only one codon. Okay, so there is no overlapping. So these are the in fact these are the general features. There are some very small exceptions in case of some of the features like overlapping. Okay, so forget about that, and just only remember these general features. First one is triplet codon, so three nucleotides acts as a single code word that codes for its corresponding amino acid. Code is next one is code is universal, so that genetic code is same for viruses, bacteria, plants, and animals. And code is without punctuation, so there is no gaps will be left between two successive codons, and non-overlapping, so genetic the codons are read read in non-overlapping manner. And the fifth one is genetic code is non-ambiguous. Okay, so this there is no ambiguity in the genetic code. So that means if you take the table that UUU codes for phenylalanine, just it is phenylalanine, nothing else, and it will not code for any other amino acids except phenylalanine. So there is no confusion at all in genetic code. So every genetic code, every code word specifies its own amino acids, its own specific amino acid. So there is no confusion at all. So one codon codes for one amino acid in one organism, and the same codon codes for another amino acid in another organism. So that kind of ambiguity is not there. Always specific codon codes its own specific amino acid. So there is no ambiguity at all. So that is fifth point. And sixth one is genetic code is degenerate. So here in the last slide, you have seen that triplet codon where three nucleotides acts as a codon to code for 20 different amino acids. So we have 64 possible codons, but still they are coding for only 20 different amino acids. So that means what about the remaining codons? So out of 64 codons, 20 codons are enough to code for 20 amino acids. So what about the remaining 44 codons? So that means here those 44 codons also codes for the 20 amino acids. So here the all 64 codons are utilized to code for 20 different amino acids. So that means codons are more but amino acids are less. So that means each and every amino acid, most of the amino acids are coded by more than one codons. So that is called degenerate. So let's quickly go back to the slide which shows the genetic code. So here if you take this example of the first one UUU, okay, that UUU phenylalanine coded by which codon UUU, but in fact it is also coded by UUC also, the second one. So that means one amino acid is being coded by more than one codon. So this is what the degeneracy. If we take, there are so many examples like this. So if we take the arginine, so you have for the arginine, in fact you have 4 codons here, codes for arginine, and 2 more codons here. So totally we have 6 codons code for same arginine amino acids, and in case of glycine we have 4 codons, and like that. Alanine, for example, we have 4, and for serine we have 4, and for leucine we have 6. Okay, so this is what the genetic code is degenerate. Okay, so more than one codon may specify the same amino acid. So there are amino acids which are coded by only one codon, and there are some amino acids which are coded by two codons. Some may be coded by 3 codons, or some may be by 4, or some may be by 6. Okay, so like that, more than one codons code for the same amino acid, that is degeneracy. And the seventh one is start and stop codons. So to start, um, we have to start the translation process, we have a starting codon, that is AUG. AUG acts as a starting codon. So the first codon of all every polypeptide chain, the first codon of the mRNA which involve in the polypeptide chain synthesis is AUG only, AUG. So that means once the polypeptide chain is synthesized from the mRNA, it contains the amino acid which is specified by AUG, that is methionine, in fact. Okay, so AUG codes for methionine. So during the translation process, just after the translation process, the polypeptide chain will be released. That polypeptide chain contains first amino acid as methionine, methionine as the first amino acid. Okay, because the synthesis of polypeptide chain starts with the start codon. That starts codon is nothing but AUG. AUG is the start codon, and like the start codon, we have the termination codons or stop codons also. So, but here we have three different types of stop codons: UAA, UAG, and UGA. So these codons are called stop codons. So if there is a codon, if the protein synthesis machinery encounter a codon from one of these three, one among these three codons, so then the translation process will be stopped. So if it encounters UAA or UAG or UGA, so then translation will be stopped. So that is why they are called stop codons. Why they are called as stop codon? They stop the translation process because they do not specify any amino acid. Okay, UAA, UAG, and UGA never specify any amino acid. And most of the cases they are stop codons and they don't have amino acids, corresponding amino acids. So if you look at the genetic code table, you will find it. Okay, here you have the stop codons: UAA, UAG, and UEGA. So these three are the stop codons and they don't specify any amino acid. So that is why they terminate the translation process. Okay, and the seventh feature is, sorry, the eighth one is genetic code is polar. Like the replication and like that of transcription, that translation is also a polar process where it always goes in a specific direction. So the synthesis of DNA takes place from 5 prime to 3 prime direction, utilizing the template in 3 prime to 5 prime direction. Template will be in 3 prime direction, and the newly synthesized the new strand that is being synthesized is always will be in 5 prime to 3 prime direction. At the same time, in the same way, RNA synthesis also takes place from 5 prime to 3 prime direction. But here, in case of translation also, the genetic code is read from 5 prime to 3 prime direction of mRNA. So that means if you have mRNA this way, so it is having 5 prime here and 3 prime here. Okay, so now the genetic code the reading of genetic code will takes place in this direction, not the opposite direction. Okay, so that is why it is polar. From always it reads from 5 prime of the mRNA, starts at 5 prime end of the mRNA and moves toward the 3 prime end of the mRNA. So that is why it is polar. And one more thing is the polypeptide chain. The polypeptide chain will be synthesized from N-terminal end to C-terminal end. Okay, so the first codon, suppose the AUG is the first codon at the 5 prime end. Okay, then the first amino acid at the N-terminal end will be methionine. So this way, so this is the head. N-terminal region is the head and C-terminal is the tail for polypeptide chain. Okay, and the last feature is codon and anticodon. The codon of mRNA can base pair with the anticodon of tRNA. So there is a specific translator molecule which is also known as an adapter molecule, which can involve in the decoding of genetic code. So genetic code means that code that translate the nucleotide sequence into amino acid sequence. So there is a specific translator in between the nucleotide sequence and amino acid sequence, and that translator is tRNA molecule. So with one end it recognizes the nucleotide sequence or codon on mRNA, and with another end it carries the specific amino acid. So that is what the codon and anticodon interaction by tRNA. So is quickly understand this here. This is AUG. Sorry, this is what the mRNA, and it is the first codon. So then now what happens? A tRNA, and this is the protein in fact, and there should be methionine, and the next codon is UUU. So there should be a phenylalanine at this position. Here the methionine. So now who is the person that can uh, read AUG and place methionine at that position? So that is the tRNA. So tRNA is the adapter molecule. It recognizes the AUG with one end and carry amino acid methionine with another end. So it will recognize AUG by means of the complementary sequence on the tRNA. So tRNA will have a anticodon sequence that base pairs with the codon. So anticodon recognizes the codon and it carries the codon specific amino acid. So this is what it is, a translator. It can read the nucleotide sequence with anticodon and it can carry the specific amino acid with the amino acid accepting arm. So that is why tRNA is an adapter molecule. So the genetic code in fact is the property, genetic code is carried out by the tRNA. So genetic code is within the tRNA. So tRNA plays a major role in the genetic code or translation process. Okay, so that is what the features of genetic code. And when we look at the genetic code table, so we have already seen the features of genetic code, and in case of genetic code table, you have 64 different types of codons. Among the 64 codons, you have one codon, that is AUG, as the uh, initiation codon. So here the AUG as the initiation. Okay, AUG, this AUG is the initiation codon, codes for methionine, and UAA, UAG, and UGA are the stop codons. So leaving these four codons and remaining all codons codes for remaining amino acids. Okay, so out of 64 codons, 3 codons are stop codons and remaining 61 codons codes for 20 different amino acids. Okay, sometimes not sometimes, so in most of the cases more than one codon codes for the same amino acid. That is called degeneracy of genetic code. And so once if we look at the flow of information, so this is the DNA sequence, that is a gene, and during the transcription we are getting an mRNA molecule, corresponding mRNA molecule. So during the translation process, what happened? So now this first one is the first codon coding for methionine, AUG, and GCC coding for alanine, and UGG coding for tryptophan, and ACU coding for threonine, and UCA coding for serine. So this way there is no gaps between two successive codons, and this is universal codon, and AUG is the start codon, and each three nucleotides acts as a codon or code word. Okay, so this is what the features of genetic code. So far we have learned about what is the definition of genetic code. It is simply the relationship between nucleotide sequence of mRNA and its corresponding amino acid sequence in a polypeptide chain, or in other words, simple words, which amino acid is coded by which codon in mRNA. So the genetic code features are triplet codon. So 3 nucleotides acts as a codon to code for its corresponding amino acid. Code is universal, so there is There is no variation in the genetic code from organism to organism. All organisms, from virus, bacteria, to higher level organisms like animals and like we, the human beings, the genetic code is same for all the organisms. Genetic code is universal. Third one is code is without punctuation, so there are no gaps left left between two successive codons. So all nucleotides are read continuously, so there are no gaps will be left, and non-overlapping. So each three nucleotides acts as a codon, and the next three nucleotides as next codon. So that means if we have 12 nucleotides in corresponding polypeptide chain, you will have, for 12 nucleotides we will have 4 4 into 3, so that means 4 amino acids. Okay, so there is no overlapping during the reading. And the fifth feature is genetic code is non-ambiguous, so there is no confusion in the genetic code table. So there are no different types of genetic code tables. One genetic code table is specific for lower level organisms, and another genetic code is specific for higher level organisms. No such kind of many number of tables, genetic code tables. Only one genetic code table that is true for all types of organisms. So that is why a codon that codes for a specific amino acid like UUU codes for phenylalanine, it always codes for phenylalanine in every organism. So there is no ambiguity and no confusion in the genetic code. Genetic code is degenerate, so that means we have out of 64 codons, 61 codons will be utilized to specify 20 different amino acids. The remaining 3 are stop codons or termination codons. So that means more than one codons may code for only same amino acid. One amino acid may be coded by more than one codon, that is known as degenerate. Start and stop codons. So you have AUG as the start codon, and UAA, UAG, and UGA are the stop codons. Okay, these stop codons cannot specify any amino acid, so that is why they cause the termination of translation process. And next thing is genetic code is polar, that the ribosomes and protein synthesis machinery, the entire translation system starts reading mRNA from 5 prime direction to 3 prime direction. So it goes from 5 prime to 3 prime direction always. So that is what the polar nature of translation. And codon and anticodon, you have codons in mRNA, and these codons are recognized by anticodon in tRNA molecule. So the tRNA is an adapter molecule that specifically recognizes a codon by its anticodon sequence, that base pairs with that and carries a specific amino acid that is specified by that particular codon. So that means if you have UUU, suppose if you have UUU in mRNA, then the tRNA will have AAA in anticodon that carries methionine. That carries phenylalanine. Okay, so that means this UUU is coded code for codes for phenylalanine. So how this is established, how the relationship between UUU and phenylalanine is established? That is because of the presence of tRNA, which recognizes UUU by its anticodon, that is AAA, that is complementary to the codon. So that is why, because of the tRNA only, the genetic code will be established. So the genetic code, the secret of genetic code is present with the adapter molecule, that is tRNA molecule. Okay, so that's about the genetic code, features of genetic code. And in the next class we are going to study how this genetic code table has been deciphered by different scientists. Thank you all.